Hello. Today in this video, we are going to be talking about the central dogma of molecular biology. And I don't know, maybe you've been feeling so far this year, we've had sort of several simultaneous trains of thought. So we've been thinking about, on the one hand, you had an assignment where you had to read about a phenotype, a disease, or a condition that could ultimately be traced back to a mutation in one particular gene. So there's one mutation in the DNA sequence that led to this very visible change in the way a person's physiology works. And then simultaneously, we've been thinking about how to build up biological molecules. So the idea of taking monomers and adding them together or connecting them together to build a polymer. And then finally, we've also been thinking about this idea of protein folding and how we start with a sequence of a polypeptide sequence of just a bunch of amino acids strung together, and how we can take those and fold them into a 3D structure that actually has a function in the body. So how a protein's function is dictated by its the sequence of amino acids that you start with. So the hope is that this will all be tied together in this with this idea of the central dogma, which means how we get from gene at the starting point to protein as the finishing point, the end goal. Instead of just jumping into talking about the central dogma, we're going to start with an analogy. And our analogy is going to be that we want to make a delicious dinner. So let's say we want to make a lasagna. And if we were to do this, our process for making this lasagna might look something like this. So the first step for making a recipe is to find the first step to making a dish is to find a recipe that you trust. Some people are really good cooks and they can just throw things together and it works out. But at least I need to start with a recipe that's going to tell me like what ingredients I need and what steps I need to follow. So we're starting with a recipe. Based on that recipe, we might copy down the recipe onto in the olden days, you'd copy it down onto an index card. Now, maybe your recipe's online, so you're copying it into a Google Doc and pasting it and printing it out. But we're copying down our recipe in some way so that we don't get our pristine cookbook dirty, or we're not taking our laptop into the kitchen, which I can speak from experience is a bad idea. So you want to keep your laptop out of the kitchen, keep your pretty cookbooks out of the kitchen maybe, and instead just bring in a copy that's a little bit more disposable. Based on that recipe, we are going to gather up the ingredients that we need to make our lasagna. So we'll read through the recipe, see what we'll need, gather up all of those things, and then based on what the recipe says, our role as the chef or the cook is going to be to combine those ingredients in just the right way, in just the right amounts, to build our lasagna at the end. So based on the way that we combine these ingredients in a particular way, we end up with hopefully a finished dish that is delicious. And essentially, this is what's happening inside of our cells to go from genes to proteins. It's a very, very similar process. So here are the differences. In the DNA, instead of a recipe book, we have our DNA. And so our DNA is basically like the best recipe book that ever existed because it has the recipe or the instructions for all of the proteins that our body might need to make. So any protein, any enzyme, any structural protein, anything that is a protein at the end, the instructions for that comes in our DNA. So it's got all of the instructions that we could possibly need for any protein in our body. But in eukaryotic cells, at least, this master set of instructions is too valuable to just be like shipping around and bringing into the kitchen, as it were. So instead, we make copies of the DNA. We're making copies of the recipes. And instead of copying it onto an index card, like what we were talking about earlier, our copy is being copied onto a molecule of mRNA. So mRNA is going to be very similar to DNA, and it's going to have all of the same instructions that DNA recipe has. But it's just a copy instead of the original instead of the original. So it might be like, you can think of if your friend missed a class and you're going to give them a copy of your notes, you might send them like a picture of your notes, instead of giving them your actual notebook that has your original set of notes. So the mRNA is just it's less valuable than the DNA, which is being tucked away and protected inside the nucleus. So the mRNA is our copy of the instructions, and then our raw ingredients are going to be, in this case, amino acids. So amino acids are the monomer to build up the polymer for a protein. So if we're making a polypeptide sequence, those individual monomers are going to be amino acids. So raw ingredients for our proteins are going to be the amino acids. And then our final product, as we said, is a protein. So our protein is our end goal for this whole process, and it's going to be what we end up with at the end. And then, just as we were saying that the chef is responsible for combining all the ingredients into a delicious final dish, ribosomes in our cells are going to be responsible for putting those amino acids together to create a polypeptide chain. So that's where the magic is going to happen, where we're putting together the ingredients and ending up with our final dish, or our final product in this case. And all of this combination of amino acid, the combining, is going to be based on instructions that we're getting from the mRNA. So we're going to be talking about this whole process in more detail, but this next slide is just going to show an overview of what the central dogma says. So the sort of sequence or the flow of how this is going to go. So we're starting with DNA, and remember that in DNA we have specific genes that are coding for specific proteins. So our DNA has thousands and thousands of individual genes, and those specific genes correspond to specific proteins. So that's why when you were reading about your disease project, or when you were reading about your disease, there was usually one gene that they could trace it back to that was mutated or broken in some way. And mutating that one gene had an effect on one protein that was therefore responsible for your disease. So there's this idea that each protein has a corresponding gene, and that gene is the instruction or the recipe for that protein. So we're going to go from DNA and then make that copy into RNA, or messenger RNA. So we're making it's got the same information, the same recipe, but we're just keeping the original DNA tucked into the nucleus, and we're going to let the mRNA copy go out into the cytoplasm where the ribosomes are. So the copy of DNA into RNA, that process is called transcription. And you can think about it in the way that, so transcription, like you might think of like transcribing notes would mean just to copy down notes, or transcribing like if you're transcribing audio into something written, you're like writing down what you hear, but it's like a literal, like word for word kind of copying. And DNA being copied into RNA is the same idea. So we have the same information and we're just copying it down into a slightly different nucleic acid, so going from DNA to RNA. So DNA to RNA is transcription, and the next step is to go from RNA into protein. And this jump is called translation. So we're literally going from one language to like a whole different language. So going from something where we're using letters and RNA, nucleic acid, to an entirely different kind of biological molecule, a protein. So that's a big jump. It's a translation of information. And in this process, we're using ribosomes to read the mRNA and use that code to put together our amino acids into a particular sequence. So that's the translation process. And then finally, these proteins are all coming together to give you a lot of the phenotypes and physiology that make you you. So your hair is made of proteins, your skin is made of proteins, your eyes are the color that they are because of pigment proteins that your eye cells make. And even something that's not visible, so something like whether you are lactose tolerant or lactose intolerant, all comes down to whether you make the protein lactase that is responsible for breaking down the milk protein, lactose. So whether you make that protein or not, whether you have a functional version of lactase that stays on past when you were a baby, that comes down to a protein too. So even things that we can't see just by looking at someone, like you can't look at somebody and see if they're lactose tolerant or intolerant, but that quality, that characteristic also comes down to a protein. So proteins have a huge effect on our physiology and our morphology, the way we look. Both of those things, huge impact comes from proteins. So on the next two slides, we're going to be looking at transcription and translation in more detail. So remember that transcription is when we're talking about going from DNA into RNA, specifically mRNA. And so maybe you remember this from ninth grade bio, maybe you don't, but our DNA is wrapped up into packages called chromosomes. But if you were to unwind our chromosomes and look at the DNA more closely, you would see that it still has this double helix structure that we know that's how our DNA is structured, right? And so if you were to look at that, you would see that there's a DNA code on both strands of this double helix. So our DNA uses 4 letters, A, T, C, and G, to and with those 4 letters, it's providing the instructions for all of these proteins that we need. So it uses just those 4 letters and it combines them in different ways to provide the instructions to make our proteins. So if we're looking at this DNA strand, you can see right here, there's a protein called RNA polymerase. And RNA polymerase is basically just protein, actually, that is responsible for copying the information from the DNA and making an mRNA copy of it. So you can see this a little bit more closely or a little bit more easily in this diagram over here, where we're starting with our DNA, and this time the RNA polymerase is being shown in green, but it's going to come into the double helix strand and sort of unzip it, and then the RNA polymerase is going to start sort of like almost like a train track, like reading along the DNA, or like when you're learning how to read and you use a finger to sort of like trace the words. The RNA polymerase is tracing along the DNA, and as it's doing that, the RNA copy is being made. So you can see the RNA copy here in red. And so as the RNA polymerase goes further and further along into the DNA, our RNA copy gets longer and longer because it's got that same information that the RNA polymerase is reading. And then finally, the last step is that the RNA polymerase will sort of fall off when it reaches the end of the gene. And then you can see that we have a finished copy of our RNA molecule here. So at the end of transcription, our finished product is this mRNA molecule that should be an exact copy of whatever information we had in that gene. So whatever recipe we needed for a protein, it was contained in the DNA, and now it's been copied into a molecule of mRNA. And in humans and eukaryotes, there's a little bit of extra processing that's going to happen at the end here. So we're going to remove some sections of the mRNA that we don't need, and we're going to adjust the ends of the molecule so that it's sort of just some final processing. But we'll talk more about that later. And instead, just suffice it to say that at the end of transcription, we've copied from DNA into mRNA the instructions for how to make a protein. Okay, so this is a diagram of the next step, which is translation. So if transcription is going from DNA to RNA, translation is going from RNA to protein or polypeptide sequence, depending on how technical you want to be. So there's a lot going on in this diagram. We're going to orient ourselves a little bit here. So on this diagram to the left, there is one component is the ribosome, shown here in green. So the ribosome is going to have 2 components. In darker green on the bottom there, you can see that there's a small subunit. So this is the small subunit in darker green, and the larger subunit is placed on top of it. So the ribosome has 2 pieces that sort of fit together like this diagram doesn't make it look like it, but sometimes you see them drawn and it looks like a hamburger bun or something, like a bottom bun and a top bun, and they're like a sandwich together. So the ribosome has 2 pieces like that, and then threaded in between the small subunit and the large subunit is our mRNA molecule. So remember, this is our final product from the end of transcription, was this mRNA molecule. And so here we can see it sort of threaded through the ribosome. We also have amino acids. So remember, the whole point of translation is that we're going to be combining those amino acids, those monomers, to make a polypeptide chain. So we need to see our amino acids, and here they're represented as little spheres or balls. And then our final player in this is going to be a molecule called tRNA. And tRNA, the T stands for transfer. So the full name of this molecule is transfer RNA. And what the tRNA is going to do is bring the amino acids to the ribosome so that they can be connected together to build our polypeptide chain. So if the ribosome is sort of the workbench or the factory where these proteins are being made, the tRNA is going to be the actual worker who's bringing the necessary materials to the work site. Okay, so this sort of introduces all of the pieces that we have, the players that we're going to be using. And if you look at this diagram, you can sort of start to see how the tRNA is able to bring the correct amino acid to the ribosome. So if you're looking here, we said that the mRNA provides the instructions to make our polypeptide chain, to make our protein. And so here's our mRNA. And you can see here in these blocks, what's actually going to happen is that our mRNA is going to be read in sets of 3 letters at a time. So this set right here, AAA, that's one set. And this triplet right here, GUG, that's another set. And then finally, this GAG right here, that's another set as well. So these sets of 3 letters are called codons. And codons are sort of the code. Codons are the key that we're going to use to read the mRNA. So in the same way that the English alphabet uses letters, but those letters have to be broken up into words to make sense, the words on our instructions are these codons, these sets of 3. So using these sets of 3 lets us have enough variety that we can use all our different kinds of amino acids. So when we were doing our protein folding activity, you guys saw that there were 21 different amino acids that we might use in our protein. And so to be able to specify, to call for those different kinds of amino acids, we need to be able to be specific. So like if there's only 4 letters, we would only be able to say there's 4 amino acids, right? Like A calls for this amino acid, T calls for this amino acid, C calls for this amino acid. So we need to combine letters into words to be able to get more variety in the kinds of amino acids that we can call for. So that's what these codons do. They allow us to have variety and use all of our 21 different kinds of amino acids. So we'll get way more into more detail for these processes, both of these, transcription and translation, when we get into the actual molecular genetics unit. Right now we're sort of in the introduction, so this is a little bit big picture, but basically what you need to know for right now is that if there's a specific codon, that codon is going to call for one specific amino acid. So in this case, AAA calls for the amino acid lysine. GUG specifies that we need to bring in the amino acid valine. And GAG specifies that we need to bring in the amino acid glutamic acid. Glutamine? Glutamic. I should know this. It's one of those. Brings in a glue. And so we're bringing in certain amino acids based on these codons, based on the mRNA code. And if this is helpful to you, then I'll explain this. But if this confuses you, then tune me out for the next minute or two. But the way that the tRNA molecules are able to know which amino acids to bring in is because on the bottom of the tRNA molecule, there's what's called an anticodon. And the anticodon is basically just going to have whatever letters are complementary to the mRNA. So if you remember from 9th grade bio, which might have been a long time ago, there are sets that there are pieces of the DNA, certain letters that correspond to one another, that like to be with each other. So G's and C's like like to bond with one another, and A's and T's like to bond with one another. And the only weird thing is that once you go to RNA, there's no T's. All of them are replaced by U's, but U's and T's are basically the same. So A's like to be with T's, which are U's in RNA, but you can see C's and G's like to be together. So G is with C, G is with C, and then again, we have an A, and instead of a T, we have a U. And then again, G and C, A and U. So by being complementary to that sequence in the mRNA, we're able to bring in exactly the right amino acid for that spot. So how this is going to work, so take that if you like it, ignore that explanation if that was confusing. But moving forward, the way that it's going to work is essentially we have a codon in the mRNA at the set of 3 letters that's going to correspond or call for a specific amino acid. So this next amino acid that's going to come in might be this cysteine amino acid. And the tRNA molecule is going to bring it to the ribosome. This cysteine amino acid is going to get added to the polypeptide chain. So in the same way that when you were building the monomers for the monomer polymer game and you were trying to build a polymer of 5 units long, this amino acid chain is growing and we're trying to add another monomer onto it. We're trying to add on this cysteine. So that's going to happen with that dehydration reaction, the same way that you guys did it before. And then now the tRNA molecule is empty. And so it's just going to leave the ribosome. And it's not charged up with its amino acid anymore, but they're very sustainable molecules. They're reusable. So this empty tRNA can just go grab another cysteine and come back and be used again. And eventually this will keep happening. So the mRNA molecule will just keep feeding through the ribosome. And each, as it goes through, it'll keep revealing more sets of 3 letters. So new codons will keep coming in. Those codons will be added, will be read, and then the corresponding amino acid will be added to the chain. And eventually there will be what's called a stop codon that will just cut the thing off, and then that means that we're done. We've built our full polypeptide chain, and we have all the amino acids that we need. And then after that, the final protein will be folded up according to those protein folding rules that you guys looked at when we were doing the pipe cleaner tape activity. So once the protein is folded up into its proper 3D shape, then it's free to go do its job in the cell. So maybe it's a pigment molecule that needs to color your hair the way it is. Maybe it's a channel protein, so it needs to let certain things in and out of your cells. Maybe it's an enzyme that's helping you break down the food that you eat. All of these jobs are taken care of by proteins, and these proteins have to be made from instructions in the DNA. We take that DNA, we make a copy of mRNA in transcription, and then in translation, we take that mRNA instruction template and we build a full polypeptide chain from it. And then once that polypeptide chain is folded up in the right way, it can go do its job. So that's sort of the big picture of how we go from gene to protein. Okay, I'll see you guys in class soon.